Hello friends, I am Harun and it's your YouTube channel Harun Biozone. Today in this lecture, I will discuss 10 very important MCQ questions from the chapter Tissue Culture, from the topic Tissue Culture. To here, so let's go. These questions are very important for clear your concept and NEET exams. So here the first question is the following. The following is a prerequisite for an artificial media used for explant regeneration. So what is explant? Explant means small part of plants that use in micropropagation. That is called propagation. That is called explant. The first option is the medium should contain source of sulfur. The medium should have very low carbon content. The medium needs to offer a carbon source. D. The medium must offer a nitrogen donor. In case of artificial or in vitro tissue culture, it is essential to take a carbon source like sucrose. Okay, so correct option is C. The medium needs to offer a carbon source. Number 2 question: what exactly is an explant? What is explant? Okay, a plant part that is grown in soil. Any plant component that has been removed and grown in a test tube. Test tube. Number C. The particular plant component produced in a test tube. And D. Leaves growing within test tube. Here the correct option is C. A particular plant part. What is explant? Sorry. Any plant part. Any plant component that has been removed and grown in a test tube. In artificial culture. In synthetic medium. The explant first discovered 1950. Explant first discover or use in the year 1950. So here the correct option is B. Next question: which of the following does not involve tissue culture? Rapid clonal propagation, stomach clonal variation, embryo rescue, transgenic plant. So what is the meaning by rapid clonal propagation? Clone means duplicate. That means any part of the plant that produce asexually or that produce same type of part are rapidly propagate. Somoclonal means plant produced from somatic cell. Embryo rescue transgenic plants means where the gene transfer from one plant to another plant to get the desired product. So here the does not involve. This question represent here embryo rescue. So what is the meaning of embryo rescue? Plant from the cells or the part of the plant that are not able to survive. What is embryo rescue? Why? Embryo rescue means help to germinate, help to develop plants plants that are normally unable to able to germinate. Okay, this is called embryo rescue. So here the embryo rescue is not a part of Embryo is part of tissue culture, but it is a part of plant breeding. Plant breeding, but not in tissue culture. Next question. Which of the following statement regarding plant tissue is false? That is, plant tissue is false. A group of cells that have a common ancestor and cooperate to carry out a certain functions. A plant tissue differ from an animal because the two have different personalities and roles. In plants, there are two different tissues type: permanent tissue and meristematic tissue. And the sclerenchyma, collenchyma, parenchyma, and epidermis are among the secondary meristematic tissue. Here option is D is false because these are not secondary meristematic tissue, these are permanent tissue. Permanent tissue. Clear? Then question: the ability of single cell to divide and produce differentiate cell in the entity is called totipotency. The ability of a single cell to divide and produce all differentiated cells in the entity, that is totipotency. Totipotency is the ability of a cell to propagate. Whole plant, whole body. This is totipotent. Correct option is totipotency. What is the meaning of multipotent? Multipotent means when a cell produce limited number of cell. Number of cell, like mesenchyme cell, neural cell. What is pluripotent? Many type of cell produce. Many type produce. And unipotent, only one type. Only one type of cell from ancestor. Here the correct option is A. Next question. What is soma clones? Soma means somatic cell. Somatic cell. Somatic cell. And somatic cell means no crossing over. No crossing over. Or no mutation, that is no mutation, that means somatic cells are no crossing over, no mutation, that is no variation occur, no variation are found. Plants chemically identical to the original plant? Not chemically. Plants morphologically identical to the original plants? Plants anatomically identical to the original plants? Plants genetically identical because there is no variation occur. So plants are genetically identical. তারপর হচ্ছে কি so the correct option is plants are genetically identical to the original plant because here variation or crossing over absent. So parents and offspring are 100% identical. That is called soma clones. What is protoplast? This is cell, plant cell, okay, and outer of the plant cell, that is cell wall present. Cell wall present. The protoplast means when the plant cell becomes cell wall less. plant cell. Cell, that is protoplast. Clear? So here cell wall plus cell wall minus plant cell minus cell wall, that is plant cell minus cell wall, that is called protoplast. So this cell wall is made by cellulose, plant cell wall, that is the protoplast. Means when the cell wall is removed, cell wall is made by cellulose, hemicellulose, pectin, lignin. So when the cell wall is removed, use cellulase, hemicellulase, pectinase এবং ligninase type of enzyme that remove the cell wall. The plant cell without cell wall is called protoplast. This is protoplast. Next question, what is meant by organ culture? Organ culture means different part of the plant, that is organ. Maintenance alive of whole organs after removal from the organism by partial immersion in a nutrient fluid. Introduction of new organs in an animal body, not with a view to create a genetically mutation in the progenies of that animal. Number C, the cultivation of organs in a laboratory through the synthesis of tissue. The aspects of culture and community which are mainly dedicated by the need of specified organ of the human body. So here the correct option is C, cultivation of organs in a laboratory through the synthesis of tissue by tissue. This is tissue culture. Number 9 question. The pair of hormones required for callus to differentiate are. What is callus? Callus is an undifferentiated mass of cell. Suppose this is explained. When the starts to grow, they first divide to produce a mass undifferentiated cell, that is called callus. This is callus. Then the right option is auxin and cytokinin. If auxin level is high and cytokinin level is low, then in medium, then form root. If auxin level is low and cytokinin level is high in the culture medium, then shoot develop first. Okay. The next question is, synthetic seeds are produced by the encapsulation of somatic embryos with sodium acetate, sodium nitrate, sodium chloride, sodium alginate. The correct option is sodium alginate. Here you can see some pictures that this is the embryo. Somatic embryo means body cell. Embryo produced from body cell or somatic cell, and this is covered by sodium alginate. Okay, this is called artificial seed or synthetic seed. These seeds are sown in a favorable environment to produce a new plant. So this is sodium alginate, and this is somatic embryos. This is the end of this video. Thank you.